Righty ho. So previous video we did the gastric phase of digestion. Now, there's actually an intestinal part of the gastric phase of digestion that I didn't really go into, but I'm about to do it now. So let's move this up here. So what we've actually got with the gastric phase of digestion is three broad phases. We've got what I talked about at the very beginning, which is before food even enters into your stomach. That is called the cephalic phase. And it is responsible for about 1/3 of the gastric acid or gastric secretions. Let's put that in bracket there. And like I said, this happens before food even gets into your stomach. Enters stomach. Then the next thing that we got is the actual gastric phase, which is broadly what we were discussing in the last video that I did. That's responsible for the other 2/3 of your gastric secretions. Let's call it acid. And that is mediated by two things: stretch and concentration of peptides. For completion purposes, we now need to talk about the intestinal part of this. So we'll call it the intestinal phase. And we're doing what is, I suppose, quite a complicated thing, really, because we're actually doing the intestinal phase of digestion, but we're actually crossing over into the gastric part. So really, this is responsible for 0/3 of your gastric acid secretion, brackets. But it actually is. And there's a reason for this. It's because when we're talking about gastrin secretion from this bit and this bit, I hope you can see my pointer on screen there. What we're talking about is gastrin secretion, and these two chaps are secreting a type of gastrin that is called G17 gastrin. And this is different because this is secreting G34 gastrin. So this one is gastrin, right? So I'll just put that back in properly. G17 gastrin, and this is called intestinal gastrin. So let's have a little bit of a zoom. Put that over there. Actually, no, let's do it like this. Let's zoom it to about 84. Move that way. And then we'll do a spot drawing. Right, so esophagus, cardiac sphincter, Follow that down. Scoop the stomach around like this. Sorry, my OCD. And finish our stomach like that. So we've got the cardia. We've got fundus. Corpus. We've got the antrum. Josie's probably going to kill me because I'm not getting these ratios correct, but I'm a physiologist, but that means this is the pylorus. And we can follow this down into the duodenum, like that. Let's colour them in so that we remember what we're talking about. And then we've got some cells that we can start to talk about. So in the corpus, we've got the cells that we were discussing the other day. These are the parietal cells. Okay, and in the duodenum, because don't forget we're talking about gastrin secretion, we've got G cells. But don't forget, these are, handy that I've left this here, secretin G34 Gastrin. Now, the G cells we were talking about previously, we were talking about secretin G17 Gastrin. I'm not really saying that they do an awful lot different in this particular thing, but we'll talk about it. Let's put a blood supply in, like this. Okay, so what happens? We take in a bolus of food. That bolus of food, here it is, comes down through the cardioesophageal sphincter into the stomach, causes the stomach to stretch, causes everything that we talked about in the gastric phase of digestion. Then we end up, after all the goodness has happened, where we get the HCl secretion, proteins start to get broken down, we get strong peristaltic waves working their way through the stomach, and they get stronger. The peristaltic waves get stronger as they come towards the pylorus. And a cool thing happens is that in and around the pylorus we can store about 30 ml of chyme. And when we get a peristaltic wave coming through this area, through into the pylorus, we get 3 milliliters of chyme pushed into the duodenum, and we get 27 mils of chyme that goes back into the stomach. 27 mils. And that is called retropulsion. So you get this churning effect. But the bottom line is 3 mils of chyme come into the duodenum. Those 3 mils of chyme will contain some partially digested proteins. Okay? These, I'll tell you what we'll do. Get rid of that. And we'll also get rid of this because we've talked about it. And it's first year stuff anyway. And what we'll instead do is we will draw in tell you what, draw it in blue instead. This blue thing represents partially digested proteins, which are called peptones. Sorry, my apologies. Peptones. Let's just do that properly. Stop being lazy. Peptones. So, we can agree that we have now increased the concentration of these peptones in the duodenum. This increase in peptone concentration is going to be sensed, if you will, by the duodenal G cells. Duodenal G cells. Now, when these guys become stimulated, what they will do is they will release their G34 Gastrin, and that G34 Gastrin will enter into our bloodstream. It will be transported to those parietal cells, and those parietal cells, as we discussed, have a proton pump that is sensitive to gastrin. So what will it do? It will put a H plus through that way. It'll take a K plus that way. And then that concentration, don't forget we got a Cl minus leakage channel that's going into there, and we've explained through acid-base metabolism how that works its way in. And what will it do? That we know will take, because we're doing this in blue, it'll reduce the pH, which will change pepsinogen to pepsin, which will then break down more proteins, increasing the concentration of peptones, and then we've got a nice feedback loop that is going on. Lovely. Okay, so that's the first bit of that dealt with. The next thing we need to talk about, because don't forget, it's not only partially digested proteins that are coming into the duodenum. We'll do this in green. We also have things like fatty acids that are coming through. Okay, this is good. We get an opportunity to talk about another type of duodenal cell. So let's just bring our duodenum down like that and put another cell in here. Now these are called S cells, secretin cells. So that's a nice, easy thing to remember. Secretin is secreted by S cells. So, the fatty acids are something that S cells are, we could say, what's the word I'm looking for? Exquisitely susceptible to. They are also, for completion purposes, susceptible to changes in increases in proton concentration as well. So as that acidic chyme comes into the stomach, let's change that proton to a black dot, just because I feel saucy, and I think we should, like that. So these S cells are stimulated by fatty acids and by protons, and it causes the S cells to secrete. They become, let's put a little plus in there, because they become excited by this. They will secrete secretin. that secretin will enter the bloodstream, and it will do a couple of different things. First thing it's going to do, it is going to find its way to cells in and around the antrum of the stomach, and this is an antral G cell. So secretin will come in and it will work its way into the antral G cell. That is going to stimulate the antral G cell. It's a G cell, therefore it is interested in gastrin. It will cause G17 secretion and release. And then what does that do? It decreases the my apologies, I confused myself then when I was talking. See, even I get this wrong and I'm making the bloody video. The secretin doesn't stimulate the antral G cell to release gastrin. It inhibits the antral G cell from releasing gastrin. So if we decrease the G17 release, we decrease the amount of hydrochloric acid in the stomach, and we therefore increase the stomach's pH. So secretin from response to fatty acid increase in the duodenum and proton increase in the duodenum causes a feedback mechanism that is an inhibitory mechanism. So therefore we can say, I suppose, so the S cells, what happens? Increase in fatty acid concentration, or an increase in the concentration of protons in the duodenum, bring about an increase in S cell secretin release, which then impacts on the antral G cells, which then decreases G17 gastrin production / release. What does that do? It decreases HCl, which then increases stomach pH. So that's one thing that it does. That is mainly, though, I would say, that is mainly in response to fatty acids. The major thing that happens when we get very acidic stomach chyme is, and I'll do this in red. So the major thing the secretin will do in response to stomach chyme, so we've done it in black with the way that it acts on the antral G cells. Now we will let's take all of this. Sorry, I just need to move something. So let's take this. Change of plan. Delete that instead. Right, where was I? Okay, so the major thing that the increase in proton concentration will do is as follows, but we need to draw in, and there's a reason I created a little bit more space for myself, is that actually we need to put in the liver, and the gallbladder. So this is liver and gallbladder. All right, so that secretin that is going through, and like I said, I'm going to do this in red. So the secretin is coming into the bloodstream, will act upon the liver as follows. So here's secretin. The secretin will cause the liver to act upon cholesterol and break it down into bile acids. Now, therefore, what we were doing by increasing the number of bile acids is we are increasing bile synthesis. Now, that bile is going to go to the gallbladder, and then it's going to come eventually back into the duodenum, via the usual routes through the sphincter of Oddi. So, that's lovely. There's something else the secretin is going to do, and that is, oh god, do a little bit more drawing. Right, let's put this here, like this. I'm drawing a nice pancreas. And that is through pancreas, like this. And my pancreatic ducts look suspiciously like my comedy alveoli as well. So I'm still doing this in red. Secretin. It will act upon the, let's put it in there. Secretin will act upon pancreatic epithelial cells. And what will happen there is it will cause the pancreatic epithelial cells to produce and release bicarbonate, which will also join the duodenum. Ah, and look at that. What have we got? This is secretin in response to an increase in proton concentrations, and we are then getting down here. Why did I do that in red? We are then getting down here. Let's create a little bit more space for myself, actually. H plus HCO3 minus. And what's going to happen then? That'll enter into a reversible reaction, H2CO3, and also into a reversible reaction where it will break down to form CO2 plus H2O. Therefore, we have buffered the increase in protons. Ah, look at that. So, what else happens? So, fatty acids. We've done. Now let's look at protons. Secretin acts upon the liver to break down cholesterol into bile acids. Which then increases bile synthesis. Which goes to gallbladder. and then eventually back into duodenum. And it also acts on pancreatic epithelial cells. Epithelial cells, which increases HCO3 minus concentration, buffer protons. Cool. So that is secretin. Okay, I'm starting to get a little bit excited by this now, because we're going to zoom in on this bit, and I'm going to draw in another cell. This time I'm drawing in an endocrine eye cell, and this eye cell is exquisitely susceptible to fatty acids. Where are we? So fatty acids, to peptones, and also, one that we haven't talked about yet, but I do fear that I'm running out of the colours, glucose. Never thought I would see myself using the glittery pen. I can't wait for Josie to see this video. Right, so what happens? Well, and bearing in mind the major response here with the eye cells is fatty acids. Okay, so just keep this in mind. As a matter of fact, I'm going to do everything in green for this. And this fatty acids causes the eye cell to release CCK, cholecystokinin. Now, cholecystokinin is going to enter the bloodstream. It's going to do a couple of things. First thing it's going to do is it will act upon the parietal cells. Don't want to do anything, right? It will act upon the P cells. It will act upon the parietal cells. It will do everything through what's called G inhibitory pathways. In other words, gastrin inhibitory pathways. That's all you've got to think of. And it will calm down that proton pump. And then, therefore, it is going to reduce the acidity of the stomach. So, okay, CCK, what does it do? CCK. It inhibits proton pump in the P cells, which causes an increase in pH of stomach. And the whole point of this is to slow down gastric emptying. Okay? Slow down gastric emptying. Hang on a second. I need to go back a little bit, don't I? Because I've done something to my diagram. There we go. CCK. There. Slow that down. Proton pump. So, what does it do? CCK decreases proton pump, increases stomach pH, and reduces the rate of gastric emptying. And this is important because basically what we're trying to do is buy the duodenum some time to deal with the amount of fat and acid that is coming its way. Okay, so that's its first job, is to reduce anything that is going to increase gastrin. It's going to dampen it down. Right. Next thing it's going to do is the bit that I think is probably the most clever. CCK will now, I need to stop touching things and moving things. CCK in the bloodstream, up there, is going to find itself a little receptor site on the liver, and it there will enhance everything that secretin does. So CCK comes along here, boom, CCK, and it will enhance all the actions of secretin. So I'm just going to move my computer if you can just hear me rummaging. The main one, and don't forget, this is because CCK is, and this is why I'm doing it in green, is that it is exquisitely susceptible to fatty acids, is to get these bile acids that we were making with secretin out. Okay, now I think it's probably worth my while doing this, changing back to black, and actually drawing in the gallbladder. I'll actually put the little red ring around it as well, like that, because that red ring is muscle. Now, what CCK will do is it will cause that muscle to contract and for the gallbladder to empty concentrated bile out into the common bile duct. And it's probably worth me pointing out something that is really super clever, is that CCK will also bind to the sphincter of Oddi and cause it to relax. So that means that any of that bile that is building up in the common bile duct is now able to get out into the duodenum. So CCK, what does it do? It decreases proton pumping activity, increases the stomach pH, decreases gastric emptying. Then, next thing it does, enhances all secretin activities, causes gallbladder to contract and the sphincter of Oddi to relax. And what does that do? More bile in the duodenum. Lush. There is something else, though, that CCK does. Let's have another little zoom. And that is, we need to zoom in around the pancreas for this. Okay. Now, CCK will also act on the pancreatic acini. So, in and around these lobes here, let's change color again, we've got a group of cells. Okay. Now if we zoom in on these, we kind of look a little bit like, let's try and do an arrow properly for a start, like that. We kind of have this effect like this, and then a group of cells like this. And if we look at a cell, then what we have is an acinar cell like this, and inside these acinar cells you've got vesicles that contain granules. So basically what we have is a binding site on these for CCK to drop onto, and then it stimulates these granules to bind, move to, and bind with the cell membrane like this, and then release the granules by exocytosis into the acini, and then they can move into the duodenum. So I suppose the question is, why is this important? Well, it's important because these granules contain the following. They contain pancreatic, and then I'm just going to list them off, proteases, amylase, lipase or lipase, depending on how you like to say it, and nucleases. And they contain precursors of these. Now it's important because what we don't want is fully active versions of these enzymes coming into the pancreas, because unfortunately that could mean you actually start digesting your own pancreas, and God knows we don't want that. That's actually what happens in pancreatitis and stuff like that. What happens is the precursors find their way into the duodenum, and then they undergo chemical changes which actually then cause them to become their active forms. Fantastic. But look at this. What is it that caused the CCK to become active in the first place? Oh, peptones, which are proteins. Tick. Glucose, well, I say glucose, any carbohydrates. What are carbohydrates digested by? Amylase. Tick. Fatty acids, lipases. Check that out. And then, of course, we got nucleases to break down actual biological components that don't fit into those categories, which is great. That's all very clever. We're enjoying this. This is good. CCK is very clever. So I suppose we better add this to our list of everything the CCK does. So what does it do so far? It decreases proton pump activity in the P cells. It increases the stomach pH, slows down gastric emptying as a result, enhances all of secretin's activities, goes to the gallbladder, causes it to contract, and the sphincter of Oddi to relax, which means more bile is going into the duodenum. Then we have to add on it, whoops, change colour. It affects the pancreatic acini cells to release just granules containing, let's list it. Granules that contain pancreatic protease, amylases, lipases, nucleases, precursors into duodenum. Whew. Okay, two more things to talk about. So let's go and come back here. I'm going to draw another cell in like that, and this time this is called this isn't let's zoom it in. endocrine cell. Okay, now the endocrine cell is going to respond to, where on earth am I going to draw this? Okay, let's just do a little bit down here. So the endocrine cell is going to be responding to distension of the duodenum. So distension from lots of chyme. Now this stretch response is going to cause an increase in the synthesis and release of two different substances called peptide Y and neurotensin, which I'll call NT. Okay, so off these guys go into the bloodstream. Whoop, like that. And they are going to affect the P cell. And guess what you think they're going to, what do you think they're going to do? They are also going to decrease the proton pump. And then that's going to do everything that everything else does. It's going to decrease the amount of hydrochloric acid. It is going to decrease the conversion of pepsinogen into pepsin. And therefore it is going to also slow down gastric emptying. Okay, so let's add them to our list. So we've got peptide YY and neurotensin, and what they do is they decrease the proton pump, which increases stomach pH, decrease gastric emptying. And these are in response to duodenal stretch because of increased chyme. All right, fantastic. Whoop. Look at that. Look at that. Right. I'm going to draw this one over here, just because of what it does. I'm not doing that for any particular reason. It's just where I'm putting it. Right. This chap isn't enteroendocrine K cell. All right, so it is. Oh god, I gotta get my glitter back out here, haven't I? Right, so, and the reason I put it here is because it is affected by glucose predominantly. Also a little bit of fatty acid as well, but just for I'm not going to swear, so I'm going to say just for kicks and giggles, I'm going to do this in glitter. Okay, so in response to high levels of glucose concentration, my goodness, look at that. It is glittery. High levels of glucose concentration, this enteroendocrine cell will release something called GIP, which stands for gastric inhibitory peptide, and I haven't got enough space to write it, so I'm just leaving it as GIP. It will enter the blood stream. And it will also impact on the P cells, and it will do all of the things that PYY and neurotensin will do, and it will do all of that cool stuff. But, and there's a reason that I've got it over here, it will also, after it's gone through the bloodstream, it will impact on the pancreas as well. Okay, and it will specifically impact on the islets of Langerhans. And in the islets of Langerhans, we know that we have got beta cells. And anybody who's done year 1 biochemistry will know that the beta cells stimulate the release of insulin. And insulin has profound anabolic effects. So everything that we have been dumping into our duodenum, and it's going to zoom out so that we can get all of this physiological goodness onto one page, which is actually quite challenging. Here we are. Look at that. So we can get, all of this stuff that we've been dumping into our duodenum. We're preparing the body via insulin into an anabolic state that we are actually going to be able to, good God, I still haven't got it all on one page. There. That we're going to be able to actually process it and get it to where it needs to go so that we can actually make use of it. Lovely. That's it. Well, I say that's it. I mean, that's it for the intestinal part. I hope you enjoyed that. I hope you found it useful.